So the theme, or my theme of this afternoon is human iPS cells as models for cardiac disease. And much of what we have learned of human cardiovascular diseases has come from genetically modified mice. But there are some limitations of mice, especially in physiology, in expression of ion channels within the heart, and mice are lacking human genetic variances like SNPs or copy number variants. So beside the mouse model, we need other systems to study human cardiovascular diseases. And human stem cell biology offers these other systems. You've already heard very nicely by Robert about human ES cells, and human iPS cells or patient-specific iPS cells are another model to study disease. I think both ways are mutually reinforcing, and in future studies for monogenetic cardiovascular diseases, the genetic mutation has to be homologously recombined, so the phenotype has to be rescued. And with human ES cell biology, you also can study the genetic mutation within another genetic background. And with iPS cells, you study the genetic mutation within the same patient-specific genetic background. So I think both models are mutually reinforcing each other for studying diseases. Since the groundbreaking work from Yamanaka now and other labs over the past years, now almost any cell can be reprogrammed in a pluripotent state. T lymphocytes is especially interesting in the light of genome-wide association studies, because biobank blood can be used to reprogram iPS cells. And then the cells can be differentiated into different lineages, as you heard in the previous talk, with different problems there, and I will highlight some within the next 15 minutes. So these cells then can be used to study in a cellular assay the pathology of a monogenetic disease. The cells, the differentiated iPS cells, can be used to screen for drugs which aggravate or which rescue the observed phenotype, and in combination with biomaterial approaches, the cells can be used for tissue engineering to study the disease in a 3D context. So in the first part of the talk, I will highlight the different cell types you're getting during iPS or ES cell differentiation in culture, and methods which you need to develop to identify really the cell you want to study. Maturation was also alluded by Robert, whether the maturation state of the cells influences the phenotype you're studying. And in the second part, I want to shortly introduce our last model for CPVT1, which is the catecholaminergic polymorphic ventricular tachycardia, and in the end I will give you a short outline. So as Robert already said, even for the cardiac system you have different cell types: atrial myocytes, ventricular myocytes from the right ventricle and from the left ventricle, and conduction system cells. In our initial studies, we used electrophysiology and the characteristics of action potentials to identify these different cell types. And we looked whether the action potential characteristics correlated quite nicely to single cell RT-PCR expression of specific markers of those lineages, like MLC2V for the ventricular line, MLC2A for the atrial line, and HCN4 for the conduction system line, and they correlated quite nicely. But the future will be that we need iPS, or we need constructs in hands to be able to genetically mark our cell types. Like, for example, an MLC2V promoter in front of a reporter gene to mark ventricular myocytes, and an MLC2A reporter for atrial myocytes. This slide here shows you still an early approach where we use this construct with the MLC2V promoter in front of a DS-RET express gene, and we infected our iPSC generated myocytes, and you can appreciate a nice co-expression of the DS-RET and the MLC2V by immunohistochemistry. But I think genetic targeting of lines will be the way to go, and with these new technologies it is now much easier to target specifically loci in human ES and iPS lines. Now to maturation. Here this slide shows you action potential characteristics over a differentiation time course. This is an early action potential after 20 to 30 days of differentiation, and this one here a late one after 2 to 3 months. And you can appreciate that they look quite different. This also goes along with different expression levels of specific ion channels there. For example, the ITO current increases there over time, which leads to a higher maximum depolarization. The IF current equally decreases over time, which leads to a flattening of the depolarization, and the IK1 current also increases, which leads to a more negative maximum diastolic potential. This slide here summarizes a gene expression profile of adult human heart, fetal human heart, and explants from 2-month iPS-generated myocytes and 5-month iPS-generated myocytes. If you look at it briefly, the 2-month more resemble fetal heart tissue, while the longer you keep the cells in culture, they are more maturing. But I mean, it is still a big issue that the cells still stay in a fetal state, and we are not able to get really adult rod-shaped myocytes in vitro until now. Okay, now let's move on to the CPVT1 syndrome. I think in front of this audience I can skip this slide only briefly. We were interested in the ryanodine receptor, and there are 2 forms of CPVT. There's an autosomal dominant form of CPVT, which is due to genetic mutations in the ryanodine receptor, and there's a recessive form due to mutations in the calsequestrin gene. And the ryanodine receptor is essential for the SR cycling of the calcium between systole and diastole. In our center in Munich, we were able to recruit 1 family, and it was a quite sad story, since both daughters were resuscitated almost a month from each other. One with the age of 12 and the other with the age of 14. And the family did not know that they were carrying this genetic bomb inside themselves. The mother was 42 here, and she never had a ventricular accident or so she never went for genetic screening. Both daughters were luckily successfully resuscitated and didn't have any brain damage, and they were treated with an ICD implantation in the end. So we screened for death genes within this family and identified in the N-terminus of the ryanodine receptor this point mutation. This is an amino acid exchange of the serine to a leucine at amino acid 406. There are three hotspots of mutations within the ryanodine receptor in the N-terminus. This is actually a new mutation here within the N-terminal region, then also in the central region and in the channel pore region. And for our study, this family agreed to take part, and we generated iPS cells from both daughters, and as a control line, iPS cells from the non-affected father. So this shows you we used for this study the classical reprogramming protocol. We used retroviruses, the 4 factors, and we got efficiently clones there, and used a differentiation protocol which was adapted from different protocols over the world, which gives us around 5 to 10% myocytes in culture. One issue was that we wanted to do single cell assays with the cells, and we didn't want these sheet of beating myocytes, but we wanted for the electrophysiology studies single beating cells, and this dissociation was quite difficult in the beginning, but we succeeded in the end. So first we looked at the essential calcium handling proteins in atrial heart tissue and in the control iPS-generated myocytes from the father and the 2 girls. And we didn't find any big differences there on protein level for expression in immunoblots. And this is immunohistochemistry from the control here on the top and the CPVT on the bottom. And you can already, this highlights again the maturation state. You don't see any T-tubules there. The ryanodine receptor is quite spread within the cytosol of the cell. So they are not completely matured, these cells. We then looked by calcium cycling, and this was a collaboration with the pharmacology at the LMU. We looked with Führer-2 experiments for calcium cycling under frequency-induced stress, and we observed 3 kinds of arrhythmias. One where every second beat was not, or where you get always these B functional action or calcium cycling. A second arrhythmia phenotype where only every second stimulus was answered with a calcium release and a completely awkward calcium cycling phenotype. And you can see here on the bottom, the more you increase the frequency, the cells with these very severe arrhythmia phenotype in the CPVT group increases compared to the control cells. We then looked for diastolic and systolic calcium under basal conditions and under isoprenaline stress. And under basal conditions between control and CPVT myocytes, we didn't see any significant differences. Only if you stress the cells with isoprenaline, you saw an increase in the diastolic calcium within the CPVT group and a reduced caffeine-induced calcium release in the CPVT group compared to the control group. We then looked further by SPARC analysis. SPARC is looking at the release of 50 to 100 calcium ions, specifically on the ryanodine receptor. And we did this in collaboration with Peter Lipp from Homburg-Saar. And under basal conditions, we didn't see any big differences between the control cells and the CPVT cells in spark frequency and spark amplitude. But what we saw is that the delay of the closing of the ryanodine receptor was delayed within the CPVT group compared to this sharp decay in the control group. This shows the statistic for what I have said right now. And then here down on the bottom, if you see these spark analysis in cardiomyocytes from iPS cells. And if we stress now the cells with isoprenaline, the CPVT cells, if you look here, control, and here CPVT, they became really crazy. I mean, the release of calcium from the SR into the cytosol under the isoprenaline stress was really enhanced in the daughter cells. And this is a statistic. You get an increase in spark frequency and also a very significant increase in the timeline of the spark in the CPVT compared to the control cells. We then looked further whether we could restore or rescue this phenotype, and dantrolene is a drug which is used very nicely for patients with hypothermia-induced skeletal. I mean, this is a complication during anesthesia. And this is due to a calcium efflux of the ryanodine 1 receptor in skeletal muscle, and dantrolene binds to the N-terminus of the RUR1, and the N-terminus of the RUR1 and RUR2 have a very high homology, over 95%. And if you look, you see for spark frequency and tau decay that under isoprenaline you get always a significant increase, which you were able to block under incubation of dantrolene. And in collaboration with the pharmacology from the TUM, Andrea Welling and Stefan Engelhardt, we also looked in whole cell clamp configuration whether we are able to see these dantrolene rescue of DADs and tachyarrhythmias. And to make a long story short, yes, we saw a rescue also in the whole cell clamp mode. Okay, so this slide summarizes the model we got from our experiments. These N-terminal mutation destabilizes or unzips the N-terminus from the central domain and leads to a calcium leak from the SR into the cytosol, and dantrolene binding to the N-terminus, and we haven't showed that, but the data from the RUR1 and dantrolene offers this solution, is able to stabilize this unzipping again. We now rescued the mutation, and we're looking whether the phenotype in these iPS cells from these two daughters is really due to the mutation. So in the summary, that's only 2 slides. I want to give you a short outlook. So I still presented you the current protocol where we reprogrammed with retroviruses and differentiated with a quite heterogeneous differentiation protocol. What are the steps for the future? So I think what is very important is that we don't have to take skin biopsies from patients anymore. I mean, we are now reprogramming from T lymphocytes, and in our hands these protocols are working quite nicely. And it also offers the advantage that one could use biobank blood for future studies. We left the retroviral system and we moved on to the Sendai virus approach, which is a non-integrating or effector-free approach, and works in our hands very nicely, offers also the opportunity for safer iPS cells, but also for disease modeling a non-integrating approach where you don't have to take out the factors you introduced in the beginning. Okay, that I already highlighted. I think it's important for future studies that the disease mutation has to be corrected, and in combination with human ES cells, one can also study the influence of the genetic background on the phenotypic expression of the disease and what is really due to the gene mutation. So I think there we will see some nice papers in the future. I think tissue engineering, I mean, if you want to study, I mean, this is only in culture, in vitro, in culture. I mean, in the end, you need to study the disease really in a 3D environment with other cells. But I think tissue engineers and biomaterial research will offer us possibilities. The hematopoietic system has the big advantage to being able to ablate the whole system and then repopulate from one cell the whole system again. The cardiovascular researchers have there a big disadvantage. I mean, it would be great if we could make a human or a rat heart within a mouse organism, but perhaps one day that's possible. Okay, and the last slide is that direct conversion or cell programming is another possibility. The neural field is really advanced there, and it gives you, with much higher efficiency in the end, really neurons. The cardiac field is still disputed, but I think it will be possible in the end also to move from T lymphocytes to myocytes or from skin fibroblast to myocytes. On the last slide, I want to thank the people within the group, especially Thomas Gudermann from the LMU for the calcium analysis, Stefan Engelhardt and Andrea Welling for the electrophysiology, and Peter Lipp for the SPARC analysis. Thank you very much for your attention, and I'm happy to answer questions.